Hi, this video will explain about the diagnostic test for COVID-19, also known as the coronavirus disease. This explanation is mostly based on the report delivered by the CDC, Centers for Disease Control and Prevention. I will add a link to the report in the description, in addition to links to other related material. First, here are some important disclaimers. The purpose of this video is to simplify the currently most common test for the coronavirus to the public. For that purpose, I discarded and skipped some steps that I found to be of less importance for the clarity of the method. Furthermore, I'm not related to the CDC nor to the WHO. I'm a PhD with a background in molecular biology and am not a medical doctor. And most importantly, this video and the information inside it are solely for your knowledge, and by no means of any health recommendation or suggestion. If you have any question or concern, please contact your local or national health authorities. Now, if you want to take only one message from this video, it will be that the genetic signature of the coronavirus is the evidence for its presence in the examined person. If we find the viral genetic signature in the person, we count the person as positive for the coronavirus. Let's go a bit deeper to the technology. So, the test is based on the technique called real-time RT polymerase chain reaction, or rRT-PCR. In order to understand the test, you need to know that the coronavirus COVID-19 is an RNA virus. RNA is a genetic sequence, similar to the structure of DNA, which is our genetic sequence. It is basically the coding sequence, the coding letters that encode for the virus information. Why are we interested in the genetic code? As mentioned earlier, when collecting a human sample, we look for the molecule as a sign or a signature that the virus is there in the sample. PCR, polymerase chain reaction, is a method that enables to target and amplify specific genetic sequences. Let's see what it means. Here is a scheme of the currently most common test. First, a sample is collected from the examined person. The next step is reverse transcription. This, in short, is the conversion of RNA to DNA. This is because the next step can work efficiently only on DNA and not on RNA. Just to remind you, if there is a viral RNA, it will be converted to DNA, while if the viral RNA is not in the sample, then nothing will happen. The final step is the PCR. Let's see how it works. As mentioned before, we want to see if we find the presence of viral RNA. Since we converted the RNA to DNA, we now look for its converted DNA. In the PCR reaction, we place many different reagents, including enzymes, salts, etc. Two important reagents are 1. The DNA sample we previously prepared, and 2. PCR primers. Primers are DNA sequences that basically act as weapon aims. They are synthetically manufactured and planned and designed to target only specific DNA regions. In our case, they target the RNA or the converted DNA of the coronavirus. Let's look at the two optional scenarios. If there is a viral infection, there is a viral genome present, and the PCR primers will target it. In that case, fluorescence will be emitted and detected, and the sample will be marked as positive for the virus. On the other hand, if there is not a viral infection, no viral genome is present, and no fluorescence will be detected. This sample will be marked as negative for the virus. To summarize this step, if fluorescence was detected, it is likely that the virus was in the collected sample. And that's it. To summarize, a sample is collected and then two main steps occur. RT-PCR converts RNA to DNA and PCR is followed. The term real-time in the title marks the ability to detect the PCR success in real time by fluorescence detection. Again, It is a very simplified explanation to the currently most common coronavirus test, and in reality, some steps are combined and of course a little bit more complex. If you have any questions relating to the coronavirus or any other health concern, please contact your local or national health authority. I want to thank you for listening. Stay home, stay safe, and stay healthy. Goodbye.